laboratory. Okay. Okay, thank you very much. Assalamu alaikum, all of participants in the in this webinar. Also, I am thank you very much for organizing this webinar. My brother Dr. Akram. My name is Fethi Ahmet Ozdemir. I am study from Bingual University in Turkey. Faculty of Science and Art, Department of Molecular Biology and Genetics. My research area plant biotechnology and plant use of this plant in the drug development. I have got three laboratory in my university. First laboratory, we will study of plant tissue culture, and this laboratory use of the aim for micropropagation and micropropagation of medicinal plant and aromatic plants and product of the every season. And also we not only product of the plant, but also product of the callus and other plant material, other plants material. We study in this laboratory, plant tissue culture laboratory, suspension culture, callus culture, micropropagation culture, also haploid plant culture and suspension culture. We aim of the in our study increase of the secondary metabolite of the medicinal plant in by the tissue culture methods. We can use of the medium like of the Murashing's cook medium, Gamborg's B5 medium, needs medium. We prepare of the medium and insert of the explant in this medium. Explant, the mean of the explant part of the plant. Firstly, we growing of the plant in the we sterilize it of the seeds and insert in the culture medium. After that, the germination, we use of this plant in vitro development plant use of the search of explant, leaf, root, node, or other part of the plant use of the explant. We insert of the explant in the culture medium, development of the cali, the shoot, root, and other part of the plant, this medium. We not use of the production this plant, and we increase of the secondary metabolite by elicitor, lack of the biotic and abiotic elicitor, biotic elicitor, jasmonic acid, salicylic acid, and other biotic elicitor. And also use of the abiotic elicitor like UV, salt, heavy metal, the temperature, pH, the use of the increase of the secondary metabolite. Also, we use of organic solvent for extraction in the this plant material extracted, obtain of the many use of the many organic solvent like methanol, ethanol, the ethyl acetate, nhexane, and other organic solvent, and obtain of the this plant material extract. We really very amazing, because when we extracted of the Kali or production in plant material in the tissue culture medium or suspension culture or root culture, we obtain of the different different compound in this in this part of the plant in medium. And extracted, obtain of the extracted, we have got a second laboratory study for macrobiology laboratory. This laboratory search of the antimicrobial activity and other microbial study. We have got a lot of bacterial strains. Gram negative bacterium, gram positive bacterium like Klebsiella, Listeria, Escherichia, or many other bacteria. We product of the bacteria in this laboratory and obtain of the antimicrobial activity result, and we will study microbiology studies. My third laboratory, cancer research laboratory. This laboratory study only focus on cancer research. We have got a many cancer cell line, like of the lung cancer, colon cancer, breast cancer, of have got a cancer cell line. We proliferate of this cancer cell line in the medium use of the RPMI, DMM12, also proliferate in the well plate, incubated of the carbon dioxide incubator. And we proliferated of the cancer cell line in the well plate after then you treatment of the obtain of the extract, plant extract. And we add of the VST1 kits in the well plates, and we waited of the one overnight, and we measure of the ELISA reader, and we obtain of this cell diet. And we can say very easily about this extract diet of the cancer cell line or proliferated of the cancer cell line. And we take of the very good result. We can take of the very good result. Every time my laboratory open of my brother Dr. Akram and student, him student, every time open of my laboratory. Also, I want that study together. I want that of the I can see in my laboratory. Thank you very much, my brother.